and then hopefully it's the right one. Is that right? The right one? Yeah, all right, awesome. Yeah, so thank you for having me today. This is very exciting to share some new research that we're hopefully coming out quite soon. So I thought I'd talk today about sort of my kind of broad interest, which is about data synthesis or integration to produce practicable, useful ecological evidence for conserving or restoring or managing ecosystems. Okay, does that work? Okay, so those of you studying Dr. Pfeiffer's Chan's course, the dynamics of coupled human natural systems, will realise that natural systems and nature are notoriously complex in form and function. So this complexity means that when we try to study it, we observe different patterns in different contexts. And when I say context, I mean like different places, time points, ecosystems, and taxa. So, for example, the effect of an invasive species on biodiversity, on plant communities, might depend on island size or something. Or another example, the effects of fire and deer browsing might depend on, have different effects on plant communities depending on the environmental gradients and mountain areas. So these studies and these effects we observe in systems are often brandished as context dependent. And this is quite difficult to deal with because with the global scale of biodiversity loss amounting to the 6th mass extinction event, we really need to implement actions locally to try and slow this decline in terms of management, restoration and conservation. But this context dependence that we observe in ecological studies really limits our transferability. That's our ability to borrow ecological knowledge or management policies from one location, one context or one taxa or ecosystem type or time frame, and effectively generalize them to another. And this leads to wasted resources. For example, if we prescribe like a blanket management action to all ecosystems based on a study's findings, it might not work everywhere because of this context dependence. And it leads to wasted resources, or even worse, to ecological surprises, so unintended consequences that were from the management intervention. So we really need to understand context dependency. Or how can we get past context dependence? So this was a question posed at the plenary at the last BES annual meeting. The question was, how can we get past it depends? And this, broadly speaking, there's thought to be like 2 ways to do this. Number 1 is using cross-study synthesis. So broadly speaking, cross-study synthesis, I mean the bringing together of results of individual case studies or field studies together and analysing variation in their outcomes in relation to contextual factors. And the other way is to do macroecology, so analysing maybe consistently collected data across broad geographic extents and to similarly shed light on why we have context dependence. So today I'm going to talk about our work in these 2 areas. One in cross-study synthesis. This paper is just in press, I think, with the ecology letters, and it's about the implications of scale dependence, so that kind of context dependence for cross-study synthesis of biodiversity. This study is from my PhD actually, so this isn't in my current post with Felix Eigenbrod. It just took me ages to finish it. And then the second one is work I've been doing with Felix Eigenbrod at Southampton, and it's about using large-scale national data sets to characterise these interactions that are responsible for context dependence. So, firstly, cross-study syntheses. Broadly speaking, these are syntheses combine the results of multiple ecological studies, so measuring the same response variable on the same response to the same driver across studies. So, for example, we might have a study that set out to measure the effect of land use change from old growth forest to secondary forest. This is a very common study conducted across the world. So how well do these happen? The question is, how well do these secondary forests support old growth forest communities? So an individual study might set out to compare the biodiversity of old growth forests, which are structurally complex, so to more recent secondary forests. And studies across the world may have different effects that have been observed. So, for example, let's look at one study that may have replicated unmanaged old growth forests and replicated secondary forests and measured their biodiversity. Maybe it's the ground layer vegetation or the insect diversity, mammals, you have it, they're interested in measuring biodiversity of these habitats. And it's thought that bringing together the results of these multiple studies can help us look at this, what is the overall effect of land use change from old growth forest to secondary forest, but more importantly, why do we have differences? Can we attribute this variation in study results, the effects observed, to meaningful factors that might be responsible for driving these differences in the studies, like for driving or moderating the effect of land use clearance? Maybe we can understand context dependence. So there's two ways that cross-study syntheses are usually done. Formal meta-analysis. So meta-analysis was a method that was born out of more experimental sciences, such as medicine, psychology, and social science. And what it does, it takes the mean differences between control and treatment groups, so old growth forest, secondary forest, maybe the mean differences, and it takes those means from each study. So on that figure on the top, so each study is a point in your analysis, and takes the mean and wants to compute an overall weighted mean that represents the overall effect of this management interventions. And then the second approach is full data synthesis. So this is becoming more common in ecology these days due to this open science revolution, where people are archiving their data, the raw data that underpins their analyses and case studies. So in this approach, mixed effects models are typically used to combine the raw data, each maybe standard forest level, replicate level data from the studies to work out this overall effect. And it's broadly akin to what's called individual patient meta-analysis in medicine, where you take the individual data, but there's different ways to do that. But this is what is most done in ecology, this use of mixed effects models. So the key issue, we have an important issue here that is quite often ignored in the syntheses, and that is that biodiversity differences that are measured between control and treatment groups are scale dependent. And when I'm talking about scale has so many different definitions, but I'm particularly here interested in the effect of grain size, so or the quadrat size at which you measure biodiversity in these original empirical studies. So it's long been known that biodiversity is scale dependent. So for example, here on the y-axis, number of species versus different plot sizes. So biodiversity will increase with spatial grain or quadrat size of sampling, and the exact nature of this relationship will depend on things like the actual total number of species in the ecosystem, the species abundance distribution, and the kind of clumpiness of spatial distribution of species within the forest patch. And here, for example, here's showing a typical comparison of old growth and secondary forest. So the secondary forests are less intrinsically rich than old growth forests because they're maybe even aged, their soils haven't had as long to develop, they've been heavily disturbed, so they have less overall species. And they also increase with sampling effort, the biodiversity of these systems. But it also follows that the biodiversity differences are scale dependent. So lots of work published by Jonathan Chase et al. Show here that yes, the actual effect size, when we're measuring this difference in studies between old growth controls and secondary forest treatments, that the size of that arrow, the difference that we observe between them, actually depends on grain size too. So at smaller plot sizes, we have a smaller difference because old growth forests are more underestimated, but at larger plot sizes, old growth forests are more accurately estimated because it plateaus after our secondary forests, and the effect size is bigger. So this is because there's the assumption that when we compare two communities, so the forest types, we standardise sampling in some way, and this is mostly done by plot size. So the idea is that if we standardise plot size, use the same quadrat size of both our habitats, then that's okay, but we're actually making an accurate comparison of the actual patch level biodiversity. But we know that that's not true based on the species area distribution. And we can instead use the raw community data and estimate the species number if the sampling was more complete, if the plot size equaled the patch size of the forest at hand. And we can estimate total richness using things like the Chow1 estimator that uses the ratios of singletons and doubletons in the community. So species density, which is the number of species per sampling unit, is very scale dependent. But species richness, which is estimated, is scale independent. So it's important to realise that when we're collating studies to combine in a meta-analysis. So what are the implications of scale dependence for cross-study syntheses? Well, meta-analyses combine studies that vary hugely in scale. If you look at, I don't know, different synthesis studies, they might say that plot size varied from between 0.0001 hectares to 100 hectares and things like that. So they combine these studies that vary very widely in scale. And then this starts to beg the question of how similar should studies be? So before you do a synthesis, it usually follows some kind of rigorous, before you get the data, it'll follow some rigorous systematic review protocol where you set out, I want to get all the studies that have compared old growth and secondary forest across the world, maybe to have a certain forest age structure, a certain ecosystem type, certain landscape types, so that what you're comparing are apples with apples rather than apples with oranges. But in reality, when you go and collect the data, you become a bit desperate because there's not a lot for your synthesis. So when you're searching, you may set out to compile studies that compare at a meaningful scale, maybe at the scale of stands, because that's what the inference you want to make after your meta-analysis. The question you're asking is what is the effect of land use change, which is at the scale of the patch on biodiversity. But these might relax when you become a bit desperate for data and you start to collect all these studies that vary hugely in scale. So this has kind of resulted in a bit of a debate in the literature about how the similar study should be. So on one hand, they should be proper homogeneous as possible. So don't compare apples with oranges. So you kind of want to compare all apples. So the studies are really similar in scale, in maybe even in climate, maybe even in the exact type of forest, if it's all conifer plantations, things like that. Because when you compute this overall effect size, this overall mean difference, biodiversity difference between your control, old growth and treatment secondary forest, we want that to be meaningful. But on the other hand, you can allow them to be heterogeneous. You can compare apples with oranges and even pears and pineapples, because as long as you shouldn't be so picky, because we can use meta-regression to attribute effect size variation to meaningful covariates. As long as we do that, as long as we recognise that the studies are different, but record all those variables and actually quantitatively try and quantify the relationship of effect size differences to these meaningful covariates. And that's probably more useful, so we can actually use that information on moderation of effects to actually target management to the right places. Okay, so what to do with this information? So as long as we actually include scale as a covariate in a meta-regression or something, or the mixed effect model, if we're doing full data analysis, everything will be okay, right? So we know that an explicit consideration of scale is probably a really good thing and will actually improve our syntheses. But it's actually not that simple. Incorporating scale into syntheses is actually pretty tricky, as hopefully we can show you here. So what are the implications, going back to this big question? Well, when you're a meta-analyst or a synthesizer or a synthesist, I don't really know what the appropriate word there is, but someone doing a synthesis, we make key decisions when we collate the data, synthesize the data, we choose a model, calculate effect sizes, all these things. And these have a huge impact on the results and the inference that we draw. So one thing is the biodiversity measure, whether we synthesize species density, that's the average species richness per sample, per quadrat or whatever, versus species richness, whether we are comparing between old growth and secondary forests, the species richness, the estimated patch level species richness, which is scale independent. Whether we choose meta-analysis using the summary effects, that's summary statistics, or the full data analysis, which uses that raw data and puts it into a mixed effects models. So if we do the meta-analysis, it's further moderated by the choice of effect size. So there's two very popular metrics for quantifying a difference between a control and a treatment group. One's called Hedges G, which standardizes by the pooled variance in your samples. And then there's a log response ratio, which is simply the log ratio between your control and treatment groups. And then the hallmark of meta-analysis is to weight studies, to give greater weight to more precise studies. And some studies choose to unweight based on legitimate reasons. So when you're calculating this mean difference, you're going to give more weight to more precise studies. That logically makes sense. However, we'll find that it has implications. So, and then also depends on whether you want to really attribute this variation in effect sizes to scale. So whether you do this meta-regression and try and quantify this difference, quantify the dependence of your effect size, the differences between control treatments on the spatial grain of sampling. For full data synthesis, when you're going to do a mixed effects model, also known as multi-level models or hierarchical models, how to parameterize them, whether to put in random intercepts and slopes, and whether, again, to include scale as a covariate and its interacting effect with your forest type, for example. So anyway, I won't go into massive detail. I will send the paper around if it's of interest to anyone. But the reason that these decisions that the meta-analysis / synthesizer / synthesis makes is because of this covariation that we get between a mean biodiversity difference, we know that's scale dependent, but also the covariance of variance with plot size and replication. So we know that biodiversity differences between communities depend on sampling grain, but what's less appreciated is the fact that variance also depends on plot size. Smaller plots generally sample fewer individuals, which can lead to greater variance. But what's even a bigger issue is that replication is usually scale dependent. So we have this logistical trade-off where we either use loads of small plots in studies or a few big ones just because of time and logistical and money constraints. And this has big implications for meta-analysis or cross-study synthesis because they change, because these big analyses use variance at various stages of the process, whether when we estimate an effect size or we do this weighting I was just talking about. So I'll just show you how. Oh, sorry, how we did it. So this study, this one that's just coming out, is based on a simulation study and empirical analyses where we just set out to just emulate these studies that compare secondary and old-growth forests across the world. So they come from populations of studies, randomly sample forest patches in step 2, and then estimate both species density, the number of species per plot, and species richness to estimate them using the Chow estimator, so that's scale independent. And then we used all these hundreds and hundreds of fake studies to conduct the cross-study synthesis using meta-analysis and full data analysis. And we know what the true effect sizes are because we put them in. So we can work out effect size accuracy and precision and things like that because we know the true values. So yes, as I said, we're looking at species density, which we know are underestimated at small plot sizes. So the effect size comparisons between old growth and secondary forests are underestimated at small plot sizes, and species richness. And we know that they are scale independent because this measure is estimated at the patch level. So we found that these decisions affect the accuracy of meta effects. So these overall meta estimates here on the left, this figure, which is the overall effect size that we estimate across studies. And it also affects our ability to detect scale dependence when it's there or when it's not there, which I'll show you here. So here for the accuracy of meta estimates, this overall mean effect, it depends on the key decisions. The analysis type, so the first 3 plots on the right show the accuracy versus this correlation between area and n. So the replication and plot size trade-off that we get, which is typical in across data sets of across ecological data sets, we have this trade-off between plot size and replication. And then I wanted to look at it in relation to that, the accuracy of these meta estimates. So these are pools of studies, about 100 each. Each of these points represents a meta estimate across 100 studies. And the first 3 are meta-analyses, and the bottom red one are these mixed effects full data syntheses. So the blues are hedges G, using slightly different estimates of the variance that you calculate, and the yellow is the log response ratio. And I'm not going to go through them all in detail, but you can just see here that there are a lot of things at play here. The take-home message is that you can get very different accuracies depending on these decisions you make, what effect size you use, the analysis type, and the weighting scheme. And it also depends on this logistical trade-off, especially if you're doing a fixed effects meta-analysis, which actually does happen quite a lot in ecological meta-analysis, even though it's philosophically unsound. And it also affects the ability to detect scale dependence. So even if you, in a meta-analysis, this means including scale or grain size as a moderator, like asking whether it moderates your effect size. In a mixed effects model, the full data synthesis, it means fitting an interaction term between forest type and scale, so the grain size. So here on the right, again, we've got the first 3 are meta-analyses, so meta-regressions, sorry. So meta-regressing the effect sizes on plot size. And on the bottom, it's this conditional coefficient that you can derive from a regression that has an interaction effect of the effect of forest type with plot size. So the first two at the top are hedges G, just with slightly different variance estimators. And you can see that, crazily, the scale dependence is stronger on the right, and it's the right that's the richness. So this is actually a scale independent measure, but it's got strong, hedges G more strongly detects scale dependence for the metric that there isn't scale dependence. So it's detecting scale dependence when it's not actually there. And that's because hedges G incorporates variance in its estimation that itself is scale dependent. So you get this weird relationship come up. The next one, the yellow one, log response ratio, correctly shows that scale dependence is stronger when based on species density, the plot density, the number of species per quadrat, and very weak scale dependence for the patch rich, the species richness estimator, like it should do. Full data synthesis does pretty well as well. However, it still has quite moderate scale dependence for the patch richness, which we know is scale dependent. And again, that is because of this phenomenon called shrinkage when you fit a mixed effects model. Go into detail in the paper and we show what's happening there. But it's because of this shrinkage when you fit a mixed effects model, you impose this normal distribution of random effects and you shrink the extreme variables based on their replication. So if you have this logistical trade-off between replication and area, similarly to a meta-analysis, in a mixed effect model, you're going to overweight those studies that are small and inaccurate. So that's the take-home message of that. So yeah, hopefully that's going to come out soon, and we propose some solutions to deal or avoid scale bias in cross-study syntheses, and that includes some R code for doing that, and kind of just ask the question whether you should really be doing a meta-analysis on like proper different studies anyway, because we know these biases become introduced. Is it really meaningful exercise to interpret these mean effects across really heterogeneous pools of study? Well, no, not really. I don't think. So we kind of make that case in this paper. So yeah, this was collaboration with some really nice people who I've worked with over the years, and it was kind of started during my PhD really. So yes, and then I said for the second half of the talk, so I think I'm doing okay for time, I was going to talk about more of the macroecology approaches. So a different kind of context dependence. So I was talking about scale dependence there, but now I'm going to talk about a different kind of context dependence, using and how we can detect it using national standardized monitoring data. And it's not about its application to biodiversity, so it's a bit different. So yes, this was a collaboration based on Physiological and broad scale forest project at the University of Southampton, and we worked with individuals at Forest Research and the Centre of Ecology and Hydrology and the Countryside Survey. So this was published last year, and it's an animated analytical framework for conducting these analyses that characterise context dependence. So it's about the context dependence that arises due to these interactions among drivers that operate at different scales. So regionally, variables that operate more slowly across space, so climatic drivers, landscape, the amount and configuration of key land uses, and more locally, those local management interventions that you do, say like thinning a forest, etc. And if we have data that span big data, lots of power, and data that span these broad environmental regional gradients, we can start to ask these questions about how these regional gradients might modify the effects of these drivers lower down in this hierarchy. It's ultimately a human construct, this hierarchy theory in these three tiers, but it's still a useful place to start. And I'll go through its application with its or demonstrate with its application to 2 case studies. So, but what the framework overall hopefully helps you to do is to characterize context dependency, understand it, what the mechanistic drivers behind it, and then usefully map the spatial variability in an ecological effect so that you can target management to the right places and where it's going to work. So I'll talk about its application that came out this year was to try and understand why there's variability in the amount of damage that's done to UK woodlands by deer, and then also some work I've been doing recently that is in prep on bamboo invasion to secondary forests in Japan. So deer damage to the UK forests. We use the UK National Forest Inventory, which is like 15,000 sampling plots all across the UK, across gradients, and it's really good data. You know, damage records are not really very subject to observer bias, and it's simply presence absence data on whether a tree has been munched at by a deer or had its bark stripped, essentially. And this is an important question to ask because deer damage is bad in that it reduces the quality of timber, and the damage to bark can open up routes for infection to treat pathogens and diseases. So a key part of the framework is about when we want to identify and understand context dependency is really using a hypothesis-driven approach. So these hypotheses concerning these cross-scale interactions, so how regional conditions might modify the effect of landscape and local conditions. So selection of deer, selection by deer of the local and landscape features comes down to the features of the wider regional conditions. And this is ultimately just comes down to this foraging theory here on the right. You know, the first, this is, what's his name? Oh man. So in the 1980s, there was a framework that was set. Jones! I don't remember the year. Sorry. Yes, this selection orders, that was a useful framework to start to try and think about these drivers that operate at different levels. So these cross-scale interactions. So we set out these several hypotheses that concern cross-scale interactions that affect the decisions that deer make in foraging. So one of these is shelter-seeking behaviour. So in accordance with shelter-seeking behaviour, we hypothesise that site selection would vary spatially with climate and deer density. So deer may select to forage and damage forest stands, more so in regions that are really climatically harsh, so really windy, wet and rainy environments that are climatically harsh. Deer may select for landscapes with higher level of forest cover, and then expected to observe stronger positive effects of forest cover on damage probability in these climatically harsh regions. And also maybe they've been shown to select for stands that have a higher stocking density that provide better shelter in these conditions. Also, deer density is known to moderate deer behaviour. They're probably more likely to shelter in less safe environments because they're prey species at high deer densities. So maybe forage elsewhere than forests. And it also depends on the availability of alternative forage. So perennial land covers in the surrounding landscape, they want to have a balance, and they like these edge habitats. So we came up with these hypotheses. And we found when we fit these generalised models using the framework that yes, the effect of surrounding cover was indeed context dependent. So on the top, we've got deer density changing from high to low to the right, and on the bottom, we've got, on the left, sorry, vertically, we've got really harsh up the top and benign conditions to deer on the bottom. And then we have it for two levels of perennial cover. So whether there's a lot of perennial cover in the landscape or it's low. And I'm not going to go through all of these again. They made sense mechanistically, but it's just to show you that, yes, the effect of forest cover, which is on the x-axis, on probability of deer damage on the y-axis, there are very different slopes there. That we can see variable effects of forest cover on deer damage according to these three variables. So yes, this is great that we can characterise this. However, it's a bit useless if we can't actually, communicate that very well to policymakers. So to do that, we made a web tool for translating the study findings. So we know that the probability of damage depends on forest cover, perennial cover, deer density, and climate. This has a policy relevant question because of the UK at the moment had all these ambitious plans to plant loads of woodland in the UK. So we're going to drastically change the landscape structure at the moment and the amount of woodland in the landscape. So we decided that was our focal variable, the management one of interest. So using this tool, if someone's interested in how the damage probability to their stands might vary, they can say where they are in the UK. Then they can change all those woodland attributes, the local drivers, its tree density, the road density, all that stuff surrounding it, which were found to be important. And then these dynamic plots will update depending on where they are in the UK by just making predictions based on the regression model that we selected. So it's kind of a useful way to try and translate this complexity to policymakers, and show how the effect of afforestation on deer damage will vary very spatially across the UK. So that's one example. And then the other example, which is, in press at the moment. No, in prep, sorry, I wish. So this is about forest invasion by giant bamboo in Japan. So giant, you might be surprised to hear this, but giant bamboo is not actually native to Japan. It was introduced in China in the 1700s, and these plantation forests were planted in these rural landscapes called Satoyama, next to residential areas. But due to widespread rural abandonment in the 1950s, after the war, lots of people moved to the cities. These rural landscapes became abandoned. So the forests weren't being managed. Secondary forests, like natural broadleaf forests, were no longer harvested for like fuel wood, and also the bamboo forests were no longer harvested for bamboo. So this meant that the bamboo started to spread into neighbouring secondary forests, and this isn't great because it inhibits regeneration, decimates biodiversity, and massively increases the cost of forest restoration, because there's recent initiatives in Japan to reinitiate management in the secondary forests, but this can't be done because of bamboo, and it's just going to keep spreading. So it's good to try and understand this spread so we can spatially target management to the right places. So we know from previous modelling studies of giant bamboo in Japan that its distributional limits are strongly temperature dependent. So here I'm trying to explain how we came up with our hypotheses that we wanted to model with these cross-scale interactions. But within these limits, within its favourable temperature limits, it's photosynthetic and growthlet. It's the observed spread into these neighbouring secondary forests, so whether it invades or not, is highly variable. So it's context dependent, and it's suggestive of interactive effects between abiotic conditions, maybe temperature, climate, and these local biotic drivers. So within the stand, maybe it's competition or facilitation at play here. So bamboo is like a seriously important crop species that's going to be really important in combating climate change, because it just grows so quickly. It can reach up to like 25 metres within a single growing season, just sucking up carbon out the air. But there's actually like not that much known about its physiology, which I find really shocking. But from what I did find, we found that from these lab studies, that typical of very fast growing species like bamboo, they're quite sensitive to photoinhibition. So that is the light-induced decline of photosynthetic activity due to excess excitation energy. And in warm conditions, thermal dissipation of excess excitation energy is possible, but in cold conditions, it's not possible. So it inhibits photoinhibition, its photosystem shuts down, and it won't photosynthesize as much and has less stores for growing. So, but also bamboo is clonal the way it grows, so it can take photosynthesis from like the mother plant. So that's why it can invade under dense secondary forest canopies. It has the capacity to do that. So we translated these lab experiments into what we might observe in the field across Japan. So under benign conditions, which correspond to either low and light intensity, or warm and high light intensity, so it doesn't have photo inhibition, we thought dense forest canopies may compete for resources and limit bamboo establishment. However, in stressful conditions that correspond to cold and high light intensity, dense canopies may actually facilitate bamboo establishment by protecting bamboo from light stress. So it protects it from this photo inhibition. So we expected to observe the shift in bamboo's thermal niche. So it's able to establish at lower temperatures only if it's under a dense secondary forest canopy. So that a dense forest canopy is necessary to facilitate bamboo's invasion and spread from bamboo forests into adjacent secondary forests. So we tested this so this is called the stress gradient hypothesis, which is classically applied to like really stressful systems like salt marshes, alpine environments, intertidal zones, but less to like classically non-stressful systems like forests. So this was one of its first applications to a non-stressful ecosystem. So we extracted the regional landscape, local variables, solar radiation, temperature, things like that, and worked out various landscape metrics, the distance to road, distance to forest edge, and the local variables, which is canopy cover, slope, and soil type. And Yes, so this hypothesis we came up corresponds to a three-way interaction between canopy cover, solar radiation, and temperature, because solar radiation becomes stressful when it's cold, and that's when canopy cover is going to be important in either inhibiting or facilitating the spread of bamboo. So, and that is what we found. We found this three-way interaction between canopy cover, solar temperature, and temperature. So at high solar radiation, where bamboo is subject to photoinhibition and light stress, dense canopies can help it to establish. So here on the left is low solar radiation, and we've got its thermal niche, so its probability of occupancy in a secondary forest along this temperature gradient. And we can see when we get to high solar radiation, this dense canopy, which is the dark green, becomes really important. It shifts. So it can bamboo can establish in secondary forests at low temperatures if it's facilitated by these dense canopies. So it's shifted. So why should we care about this? What are the management implications? Well, as I said earlier, recent initiatives in Japan are to revive traditional management of abandoned secondary forests to enhance biodiversity all across Japan. And some work we did last year, this again was from my PhD, it was a meta-analysis of management effects on biodiversity in plantation and secondary forests of Japan, found that unanimously thinning forests would increase biodiversity of loads of taxonomic group, ground layer plants, invertebrates, mammals, everything. So it's recommended in the national biodiversity strategies to thin these to revive management, and there's lots of like incentives from the government going to local groups to revive management at the moment, specifically in urban areas, like with the Satoyama initiative and things like that. So it's good for biodiversity. However, we know that it's got this removal of canopy cover through thinning is going to affect bamboo differently in different areas of Japan. So we can predict the likelihood of bamboo invasion into adjacent secondary forests across Japan at 100% canopy cover. Then a classic canopy cover reduction by thinning is about 60%. And again, we can predict the likelihood of invasion at those ones. We simply look at the difference, the effect size, or the risk difference. That's going to represent the risk difference is going to represent the change in the likelihood of bamboo invasion. So it really characterises geographic variability in the magnitude and, in this case, the direction of forest thinning. So it ranges from blue to red, which is a positive and a negative impact on the likelihood of invasion. So applying this blanket management prescription of thinning to enhance biodiversity, it's going to have very different effects on bamboo invasion. So we should really target monitoring to certain areas in Japan where this thinning is also going to increase blue areas, where it's going to increase the likelihood of invasion in these warmer areas of Japan. So yeah, in cooler areas, thinning will reduce the likelihood of bamboo spread, and in warmer regions, thinning may increase the likelihood. So it's important that management and monitoring of it is spatially targeted. So the take-home message, I guess, of this talk is let's not try and get past or account for context dependency, but let's embrace it and really try and understand it, like the mechanisms going behind this with, hypothesis generation. When we do syntheses, we should really be aware that syntheses can introduce new kinds of biases. It really amplifies those within studies when we start to apply syntheses across studies that maybe favour precision over accuracy and things like that. That's not actually desired in a synthesis, but it is what they ultimately do, because they're from medicine, which were based for very simple study designs. It really requires hypothesis-different approaches, but we do have the literature. We have enough literature now that exists out there for us to come up with these hypotheses that determine these complex interactions in nature. And we need to communicate these context dependencies in practical ways that can be used by policymakers, like with making these maps and tools and things like that. So that is it. Thank you very much for listening.
Thank you, Becks. That's very interesting. Questions?